All right. Thank you for everybody who's joining us today for this monthly Flow Friday series, which is a pretty new series, and I'm really honored to be invited to talk a little bit about multiparameter flow cytometry in hematology and immunology. I wanted to break down today's talk into half the portion will be more technical in terms of execution in using flow cytometry for a lot of the experiments that we do today in immunology and hematology. And then the second half, we'll talk a little bit about data, biological discoveries. So without further ado, so in terms of technical considerations for high parameter or multi-parameter flow cytometry, I think that there are three biggest components when designing these experiments. First and foremost would be the experimental design, followed by if you're doing a sort, that post-sort recovery is pretty crucial for the assays downstream. And then finally is if you're doing NGS applications, the data record and inventory becomes very pivotal because these experiments usually stretch for a pretty long time. So I'll talk a little bit about some of the experiences and tips that I have learned and adapted along the way in the last few years. So in terms of experimental design, typically, we do analytical flow cytometry to start with, and then we kind of derive the sort strategies to sort and validate the cells functionally. And some of the considerations when you're planning a sort pretty much circles around, what is the target application downstream? Are we doing a culture? Are we doing a sequencing? Or are we doing multi-functional assays where we have to break the cells into multiple applications? And that dictates how we back calculate a number of cells to actually use for a sort. Typically see students really struggle with this one when we teach. And the next one is, people typically sometimes don't consider that the sort efficiency is not going to be 100%. So I'll talk about some of the strategies we can use to actually make the sort go more efficiently and help with the recovery. And then finally, when we're planning an experiment, the duration of an experiment sometimes, can be quite challenging to predict. So I'll talk about how I usually calculate that as well. So target application, like I mentioned, if you're doing NGS, then it becomes a single cell or a bulk approach. And then we also have to talk about, are there many manipulations we're going to do after the sort? And then again, on the topic of next generation sequencing, the goal of the NGS application also dictates what we're going to do. Because in hematology, especially, a lot of times we're studying a relationship between cell types. So one progenitor to another progenitor, or a multipotent progenitor to bipotent progenitor to unipotent progenitor. But in some of the other cases, we're doing really targeted gene expression and chromatin data mining. So for example, if you're really interested in one specific progenitor type, you might only want to limit the assay to focus on that cell type. And those will actually change your approach drastically. Now, a little more about the difference between a bulk RNA sequencing and single cell RNA sequencing before we proceed further is obviously, one is on a single cell resolution, the other one is not. And then mainly for flow cytometry is concerned, you're talking about the different number of cells as input. So for a bulk assay, you would need somewhere between 100,000 to 500,000 cells as input. But for single cell, if we're just dealing with RNA, you could proceed with as low as 5,000 cells. So that's, almost like a 2 to 10-fold difference here. The other thing is, post-sort manipulation. So if you're doing additional manipulations on the cell, such as, doing additional staining of antibodies for like a site-seq approach, or if you have to permeabilize the cells or lyse for nuclei-based assays, then you're accounting for even more losses after the sort. So that also has to be accounted for prior to executing the experiment, because you will need to jump up the number of cells you use as input for the assay. Now, like I mentioned a little bit, right, for the goal of NGS in the context of hematopoiesis, if you're studying a relationship between the cell types versus targeted gene expression, as I illustrated here, in these cartoons on the bottom. So if you're studying a relationship between cell types, right, the goal is to capture that bridge and that gap between the cells and not lose anybody along the way. But then for targeted gene expression or chromatin data mining, you want to make sure that the sort is as pure as possible. So it's very focused. So, I would say, a lot of times we say that you have to think to the end of the experiment and think about what is the goal of doing this thing before we go ahead and really come up with either a purification or enrichment strategy for the sort. So again, to summarize, right, that is really focusing on, do you need to include the edges and the gaps in the cell types or not? So all that being said, the next part would be the back calculation of number of cells that needed to serve as the input for a sort. So usually what I use is this kind of universal formula where, let's say I'm targeting for 100,000 cells, then I'll divide that by the percentage from the flow. So for example, the target cell population is 1% of total input cell type. Then I multiply that by this 0.5. So that's really accounting for like 50% of cell loss. So that could happen either during the washes, the staining, the sorters, sort efficiency may be 70 or 80%. And of course, this factor would be changed based on the operator. Ideally, you want to shoot for like 0.8 or so to account for 20% cell loss. But that may take some practice and experience. So since this lecture is recorded for educational purposes, I figured that I would like to put this out there to help some of the newer operators out there who are just starting to get used to these assays. Now, I keep talking about this idea of sort efficiency because like we all know that you will not recover 100% of the cells that you start with. That is impossible. So in some cases, enrichments could really help enhance that sort efficiency. So in hematopoiesis and in the mouse system, for example, you could do a c-Kit or what we call CD117 enrichment prior to the sort if you're studying progenitors and stem cells, because you know that all the stem cells and progenitors must express CD117. So then in this case, enriching for c-Kit, which is only 2% of total bone marrow, will drastically increase my sort efficiency because instead of sorting on 80 to 100 million cells to get my target stem cell and progenitor population, I can just sort that 2 to 3 million cells to really drastically shorten the sort time and increase the sort efficiency. Now, in the beginning, I said that, another key consideration is how long is this experiment going to take, because that's also going to dictate your downstream application. So that, we will talk about, right? Like on the sorter, you cannot sort too quickly because that will increase your rate of having a clog and collision events as well. But then if you're too slow, obviously the sort will take just forever. So usually I would say that you want to sort at less than 4,000 events per second. And then to sort 10 million cells, that would take about 45 hours to an hour, 45 minutes to an hour. Of course, you have to account for the setup as well. So that's how I typically consider experimental design when it comes to designing a sort experiment for downstream NGS or culture experiments. Now, a lot of times we say, okay, everything else has been pretty well planned, but then my cells are not very viable after the sort, where I didn't recover as many as I thought that I could. And most of the time, I think that these problems fall into 3 categories. One is your collection buffer, so the sorting collection buffer, and then the collection tubes, and then lastly, the handling temperature. So in terms of a sorting buffer, sometimes the cells are really sensitive to serum, so you can't actually quite use FBS. So then in that case, you have to resort to BSA. But then in our experience, usually if you use like a higher percentage of FBS or BSA, it serves the myeloid cells pretty well. Now, if you're dealing with like T cells and B cells, for example, then you want to avoid using a PBS-based buffer supplemented with FBS, but instead using something like HBSS supplemented with HEPs and make sure the pH is correct. Now, another thing is if you're dealing with like a low number of cells post-sort, I would highly recommend that you use like a DNA low bind tube to account for that less of a loss during the recovery process, because the cells tend to stick onto the walls of the tube. And then if this is not for a culture assay, then I would say don't even autoclave the tubes, because that will also increase their stickiness. The other thing is, I think a lot of times when we're handling the cells, the general practice recommends that you should do everything at 4 degrees. But then if you're dealing with something like neutrophils, putting them on ice at 4 degrees is actually a very bad idea, because you will increase the neutrophil cell death drastically. And the other thing is, like I would not recommend that you switch between like a room temperature to incubation to 4 degrees back and forth. It is better to remain the same temperature to avoid that temperature shock for the cells to preserve their viability. So that pretty much summarizes it for the experimental design portion. And then I wanted to just talk a little bit more about the post-sort recovery in addition to what we have already discussed. So I think sometimes we think about centrifugation and then we're not thinking about during that centrifugation process, the cells could also drastically die because they're very fragile. So usually post-sort, especially at a pretty pure population of granulocytes such as neutrophils, I would always centrifuge them at a lower speed and extend the duration to pellet the cells. And the other thing that I have seen is cell death induced by pipetting. Because in really thin pipettes, you actually put a lot of pressure every time when you pipette the cells up and down. So then using like a wider tip pipette would greatly help to preserve the cell viability in that case. And finally, for NGS applications, especially, the data record and inventory is very crucial, because sometimes, by the time you get your sequencing data back, it's been like 2 to 3 months since you did the sort. So I would always say that I recommend that you export a PDF with the sort stats to backtrack for the experiment. So that way, when you sit down with the bioinformatician for the data analysis and something doesn't look quite right, you can immediately go back with the experimental date and setup and the number of events that you got to account for any possible problems that could have happened along the way. So that summarizes the technical portion of this talk, which I think is crucial to set yourself up for success. So now with all these practices in mind, we can talk about some of the data in biological discoveries that I have done in the last couple of years. So I think that, the biological discovery that I have done really addresses two topics. One is the high parameter flow cytometry design, and how could you leverage some of the existing tools to help really build the complexity of the data. And the second thing is, how do you leverage the batch effect and human-to-human variation as we start to see a lot more human studies coming out with pretty large sample sizes. So in my PhD lab with working with Lee Grimes and Nathan Salomonis, along with their postdoc, Sean, we have built a pretty big CytSeq human bone marrow atlas. And this is using a CytSeq approach. So you not only get the RNA, you also get the antibody information from sequencing. Now, we wanted to parallelize that with real-world flow cytometry data. But as we all know here, that it is really impossible to do flow with 133 antibodies with our existing analyzers. So in this case, right, we have to think of a way where we can resort to an approach that would allow for that real-world flow cytometry data coming from this many antibodies. In this case, we chose the tool called Infinity Flow, where you have a backbone panel and you put all the rest of the markers of interest on the same channel. And then so in every single tube, you'll have the backbone plus, for example, CD1 on PE. Then the next tube, you'll have backbone plus CD2 on PE, so on and so forth, to achieve that imputation, where in the end, you get this one, what we call infinity object from all the antibodies of interest. Now, usually in human bone marrow flow cytometry, we look at the data after live singlet gating at CD45 versus site scatter to gate for the general populations. So in general, you should see granulocytes, monocytes, lymphocytes, and like a blast population, which should be quite small in healthy human donors. Now, this population will be blown out of proportion when it comes to like leukemia samples. And since we acquired primary human bone marrow, we also expected to see a CD45 negative, primarily stromal population, which we do in this case. And then here on the right side is just a UMAP representation of all these cells that I have gated on the left. And then as you can see, that the major lineages are all present and they're connected, which is a good thing, right? Because these cells are related to each other. However, one thing that I have noticed is when we just put all 9 donors that we have sampled together in a very simple UMAP, so the experiment is done the same way for every single donor, and every single color represents an individual donor. You can see here that every single color is occupying its own space, indicating that, you know, if you just look at this UMAP, you would think that all 9 donors have completely different cell populations. And we know that that would be impossible because we sampled 9 healthy donors and all these major cell types should be present in every single donor. So that indicates a pretty significant batch effect between the experiments and therefore between the donors that need to be addressed before we can even start to think about the human-to-human variation or consistency, depending on which way you look at it. So to do that, I resorted to another known tool called Cycombine, which was published last year. And it really allows for this robust integration of cytometry data sets and, more importantly, batch effect correction. And the batch effect correction is done by using a pre-existing tool, another one called Combat, that's been around for some time. So by using this tool, you can see that after the batch effect correction on the right side, the majority of the cell populations from all the donors are overlapping. And this is very good, right? Because it proves that not only did we have batch effect, the batch effect can be corrected for using this tool. And after we use the tool, we can see the alignment of the shared cell types, indicating that, you know, the donors that we have sampled indeed fit what we have expected, which is to have most of the cell populations present. Now, when we talk about batch effect correction, and in this case, it is antibody level data, we oftentimes have to think about, you know, did the correction make sense? So in this case, I examined the data using this is kind of like a stratified histogram plotting approach. So if you're used to flow cytometry data, this should look pretty familiar. And then the red color would indicate post-correction, and then the blue is pre-correction expression data of each marker. So here I'm giving you 2 examples. So on the left is this marker CD4, which we all know marks for a population of T cells. And as you can see here, each row represents a different donor. There are 9 donors in total. And for CD4, there's really not much correction that needed to happen. However, for some of the other markers, such as CD29, you can see that even though for some of the donors, the correction may not be as significant, for the other donors, the correction is actually quite severe. And then this correction, as you can see here, I'm taking this donor 2 as an example, previously had maybe 3 peaks that you can distinguish, but these 2 are sitting really closely. The correction actually stratifies the data without losing that modal resolution and modal distribution. So this indicates that to us, the batch effect correction worked, and it works differently for each antibody as we expected. Now, by examining all 133 markers that we have done on all 9 donors, we found that there are 40 out of 133 markers that show similar expression patterns across the 9 donors. And this really goes to show that, you know, we never really had a study before that addressed this donor-to-donor variation in flow cytometry. which actually, in my opinion, should be leveraged for future studies because not all markers are going to show a similar expression pattern. And therefore, some of the cell populations that you are able to gate and see in one donor may not look the same in another donor, but it doesn't necessarily mean that that population is not present. So I think some of the other considerations should be how do we leverage that information and seek that consistency of cell types that are present between the human donors, given the variation. So this is a short talk. In summary, the first part I talk about how there's a lot of technical considerations that need to go into the experimental design and the execution of techniques. And then I touched on how we can use a combination of bioinformatics tools and flow cytometry to help us get that desired complexity of real-world flow cytometry. And lastly, I also talk about how we can use existing tools in a new approach, which is to really correct for that human-to-human sample acquisition batch effect, and therefore addressing that true sample-to-sample variation. And some of the existing approaches can help tremendously, as we have seen here today. And I think going forward, a lot of the times when we acquire the data, it is worth the time to sit down and think of ways to utilize existing tools to help us stratify the data and get more out of every single experiment. So that would end my talk today. Like I mentioned, this is a short talk, but if you have any questions, feel free to jump in. Thank you. Okay. Thank you guys so much for your time.